Good morning, afternoon, and evening. Thank you all for joining today's webinar to learn more about mycotoxins, environmental enteric dysfunction, and inflammation, and their implications for research and programming on child growth and nutrition. My name is Katie Appel, and I'm an assistant researcher for the Feed the Future Innovation Lab for Nutrition and will be your MC for this webinar today. As attendees are joining, I'll begin by going over some housekeeping items. I'd like to direct all attendees to a few functions on the Zoom webinar. At the bottom of your screen, you should see a chat icon and a Q&A icon. Use the chat feature to engage in relevant conversation with other attendees. If you have a question for one of the panelists, please use the Q&A feature. Panelists will respond to questions in the Q&A box throughout the webinar, and we've allotted the final half hour of this webinar for Q&A. If you're experiencing any technical difficulties, send a message in the chat box to all panelists so that our technical support staff can work with you to resolve them. This webinar is being recorded and will be made available on the Innovation Lab for Nutrition website and the USAID Advancing Nutrition website. There you can also register for upcoming webinars and view recordings and slide decks of previous webinars. We will repeat these technical housekeeping items in the chat throughout the webinar as people may join in at later times. Before I introduce today's moderator, I'll give a brief introduction to the Nutrition and Innovation Lab and our webinar series. We are a Feed the Future Innovation Lab supported by USAID Bureau for Resilience and Food Security, and we are active in supporting research and capacity building to build the evidence base around critical questions linked to agriculture, nutrition, and health. As you can see from this map, next slide, we are active in Sub-Saharan Africa and South Asia. More information can be found on our website at nutritioninnovationlab.org. Next slide, please. The Nutrition Innovation Lab is a consortium led by the Friedman School of Nutrition Science and Policy at Tufts University with US university partners, including Purdue, Harvard T.H. Chan School of Public Health, Johns Hopkins School of Public Health, and Tuskegee University. In addition, we partner with government agencies in our host countries, UN agencies, local and international NGOs, as well as universities around the globe. Now it is my pleasure to introduce the moderator for today's webinar. Dr. Ahmed Kablan is a senior science advisor in the Center for Nutrition, Food Safety Division, Bureau for Resilience and Food Security at USAID. He manages several research programs in nutritious and safe foods, including the Nutrition Innovation Lab, Food Safety Innovation Lab, and Post-Harvest Loss Reduction Innovation Lab. Dr. Kablan has a Doctor of Pharmacy degree from Jordan University of Science and Technology and a PhD in Cellular and Molecular Biotechnology from the University of Bologna in Italy. He has worked for over 20 years in human nutrition, international development, science policy, and regulatory research, and is a well-published scientist. In 2012, Dr. Kablan was awarded the American Association for Advancement of Science, Science Policy, and Technology Fellowship. Dr. Kablan, over to you.
Thank you, Kate. Appreciate the introduction and welcome everyone. Good morning, good afternoon, or good evening. I know we are being joined with audience from all over the world, and it is my pleasure to moderate this webinar from the Nutrition Innovation Lab on mycotoxin environmental dysfunction and inflammation. And as you can see from the title, it goes through a whole spectrum of topics that are related to several aspects of nutrition, from food safety and water and environmental safety, and the impact of these different factors in the overall health and possible pathways these factors could contribute to nutritional outcomes. As the new research has continued to emerge that reinforce the integral role for food safety and food security and nutrition, USAID recognized the need to build upon previous investment in food safety and elevate the priority of food safety and nutrition within USAID. In the past year, or in 2020, we were excited to announce the establishment of the first ever food safety division, which is housed within the Center of Nutrition, and that housing or location is intentional to highlight the importance link between food safety and nutritional outcomes. Prior to the establishment of the Food Safety Division, USAID has invested heavily in several research related to food safety, including aflatoxin research and the reduction of post-harvest losses via several of our innovation labs, including Peanut Mycotoxin Innovation Lab, Post-Harvest Loss Innovation Lab, and of course, our flagship research and nutrition program, the Nutrition Innovation Lab, that invested significant resources and time in researching the linkages between aflatoxin or mycotoxin and child development outcome and birth outcomes. The Food Safety Division was established to build upon this work and to address the heavy and undervalued health and economic consequences of poor food safety practices in developing countries. While the food safety divisions, as well as many of other food safety investment launched in the midst of COVID-19, the global pandemic had underscored the importance of addressing food safety to achieve food security and nutrition. One thing that we need to keep in mind to address food safety from all aspects, whether we're looking at mycotoxins, environmental factors such as wash or poor food safety hygiene and water hygiene that lead to environmental dysfunctions, we have to think about from a systemic approach. And that is very important to think through a food system linkages, how we can achieve and reduce food safety factors. I am so pleased to be with you today and introduce our speakers that will talk about not about what we have presented in the previous webinars under mycotoxin environmental dysfunction inflammation, but what are the implications of this for research and programs on child development nutrition going forward. What we know, what are the gaps or potential research opportunity to expand our knowledge as we are exploring and trying to establish programmatic and policy guidelines in order to have more effective actions toward reducing food safety and nutrition impact. We have on our panel we have Professor Christopher Duggan is a pediatric gastroenterologist and a nutrition physician at Boston Children's Hospital, where he directs the Center for Nutrition. He is also a professor of pediatric and a director for the Division of Nutrition at Harvard Medical School, and a professor in the Department of Nutrition and Global Health and Population at Harvard T.H. Chan School of Public Health. Welcome, Professor Duggan. We have also with us Dr. Shibani Ghosh, is the research associate professor at the Friedman School of Nutrition Science and Policy. She's also associate director for the Feed the Future Innovation Lab for Nutrition, with experience working in the Middle East, Sub-Saharan Africa, and South Asia, and have the pleasure working with Dr. Ghosh for over 8 years now. Welcome, Dr. Ghosh. And finally, we have Dr. Patrick Webb. Professor Webb is Director for Feed the Future Innovation Lab for Nutrition, the Alexander McFarland Professor of Nutrition at the Friedman School of Nutrition Science and Policy, and the Principal Investigator of USAID Food Aid Quality Review Project until 2005. Professor Webb was the Chief of Nutrition at the United Nations on WFP. Welcome, Dr. Webb. And again, I had the pleasure of working with Dr. Webb for over 8 years now. Next slide, please. And now we'll start with Dr. Christopher talking about the first talk, mycotoxin environmental dysfunction and inflammation. Over to you.
Well, thank you very much, Ahmed, and good evening and good afternoon and good morning to all of our attendees at the seminar. It's really my pleasure to join this august group and discuss with you and our colleagues about the different roles of these different concepts. I will be focusing mainly on recent studies in our group and others that have addressed and defined this concept of environmental enteric dysfunction. Next slide. I have no financial disclosures to make. I do want to explicitly thank my colleague, Dr. Jackie Lauer, who led a similar topic on a Nutrition Innovation Lab seminar several months ago. Dr. Lauer, who's now at Boston University, has pioneered many of the assays that I will be describing in the assessment of environmental enteric dysfunction. Next slide. In 2008, a very influential review article was published in The Lancet by Zulfikar Bhutta and colleagues in the series of papers on maternal and child undernutrition. And as the title of the paper points out, the focus of this publication was on identifying and highlighting specific and well-tested interventions to improve both maternal and child health. And they're listed in the table on this slide, which I won't review, but includes, as you can imagine, a number of different nutritional sensitive and nutrition relevant interventions, as well as improvements in infectious disease therapies and preventions. Next slide. This modeling study went further and evaluated how many deaths and reductions in stunting could be achieved by the global community if both the general nutrition, micronutrient interventions, and disease control interventions could be addressed throughout low and middle income countries. And in the lower half of this table, you can see that furthermore, the authors hypothesized that if coverage was low at 70% of all of the interventions, moderate at 90% of all interventions, or very high at 99% of all the interventions, there was a concomitant reduction both in deaths before age 12, 24, and 36 months, but in the reduction in the prevalence of stunting. And that's the focus of our topic today, which is childhood growth. And next slide, you can see that if 99% of coverage with all of the interventions summarized in the previous table, only roughly a third of childhood stunting could be prevented. And that really caught people's eye to say, well, if that's all we know, what explains 2/3 of the effects of childhood stunting? How can we investigate other factors that might be related to childhood stunting? Next slide. So let me turn to the idea that living in poor communities with high rates of enteropathogen exposure might be related to childhood stunting. And this was an ingenious study published by Francis Nguri, Rebecca Stolfutz, Johns Hopkins University, and colleagues in Zimbabwe. And what these groups did was visit 21 households in rural Zimbabwe, and 2 observers spent 6 to 8 hours per day to examine how feces were disposed of and how people were performing in terms of wash interventions and sanitizing and cleaning their hands before and after meal preparation. Next slide. Table 5 of this paper points out that half of the people were disposing of feces in a garbage pit close to home. Next slide. In addition, the investigators cultured 4 E. coli, a common enteropathogen and resident in the human gastrointestinal tract and animal gastrointestinal tract. They evaluated whether food, water, skin of the woman's breast, hand swabs, kitchen floor included E. coli. Next slide. 50% of the caregiver's dominant hand cultured positive for Escherichia coli. Next slide. 73% of the kitchen doorstep was positive for E. coli. Next slide. And not surprisingly, 100% of the chicken feces that were found in and around the house cultured positive for a bug that's known to be in the gastrointestinal tract of humans and animals. Next slide. In addition to testing the fingers, food, and drinking water of infants, these observers noted that children were eating handfuls of soil, and 2 ingested actual chicken feces a couple times in the 6 hours of observations. And the calculation would be that if you eat 1 gram of chicken feces a day, they would expose their gastrointestinal tract to up to 23 million copies of E. coli as a species. So confirming in an ingenious observational way what we know intrinsically, that children who grow up in poor communities, next slide, unfortunately communities that look like this, that have inadequate resources for waste disposal and use of good drinking water, have problems with their gastrointestinal tract. Next slide. So the concept of environmental enteric dysfunction is not a new one. It's actually a concept that has been, that was discussed in the 1970s among US Peace Corps volunteers who were returning home and were found to have a reversible cause of enteric dysfunction, or sometimes an enteropathy or a lesion in their gastrointestinal tract. Indeed, a few years ago, a group put together a diagnosis or diagnostic description of this concept of environmental enteric dysfunction. And from a gastrointestinal standpoint, it's helpful to review what the normal small bowel histology looks like under the microscope, which is figure A. You can see that the reason the gastrointestinal tract has such a high capacity for its two functions, namely gastrointestinal absorption of nutrients and a barrier function to prevent bacteria from getting access to the bloodstream. Those functions are improved by having very nicely developed villi and crypts to increase surface area. With environmental enteric dysfunction, however, next slide, there is a distinct blunting of those nice finger-like projections into the bowel and an inflammatory infiltrate in the lamina propria of the tissue. Next slide. Indeed, we conceive of environmental enteric dysfunction as a subclinical condition. In other words, people, young children and adults may not have symptoms of gastrointestinal inflammation or infection, but they can have important alterations in mucosal barrier function and absorptive function. Histologically, this consists of, as noted in panel B, atrophy of the villi, hyperplasia of the crypts get longer, there's increased intestinal permeability, there's an inflammatory infiltrate, and we'll talk about systemic and gastrointestinal inflammation later in the talk, and malabsorption of nutrients. And the reason the group really didn't call environmental enteric dysfunction as environmental enteropathy is that it's clear to me that even in the absence of histological changes, in the absence of true changes in the pathologic specimens of the small bowel, there are functional changes that can occur. Next slide. I think we can still say that the exact cause is unknown, but it's seemingly likely results from chronic fecal oral exposures for enteropathogens that occur in poor sanitary conditions and in low and middle income countries, as illustrated by the data from Zimbabwe. Next slide. One of the advantages and disadvantages, I guess I would say, in the field of environmental enteric dysfunction is that a number of different biomarkers have been proposed to diagnose the condition, because obviously sampling of the small intestine is often an invasive procedure, although I will say that less invasive approaches to sampling have been and continue to be developed. But because the gastrointestinal tract, as I said, its role is both to absorb nutrients as well as to keep toxins and bacteria out of the body, it's not surprising that among the biomarkers include measures of intestinal absorption, as noted in the column B, as well as measures of mucosal permeability. Other markers, including those of enterocyte mucosal surface area, gastrointestinal inflammation, and microbial translocation, have also been proposed as markers of environmental enteric dysfunction. So that's the good news, is that there are a lot of different candidate biomarkers. Next slide. The disappointing news is shown in this slide, which is a bit confusing with a lot of numbers, but let me back up and just say that these are data from Rebecca Campbell and Parul Christian in colleagues in rural Bangladesh. And in the setting of a cluster randomized trial of complementary foods, they evaluated measures of environmental enteric dysfunction at 18 months of age in children who had participated in their trial. What you can see here is a relatively poor correlation. This is the Pearson correlation coefficient among a variety of different candidate biomarkers. The top line, the top row, is the lactulose mannitol ratio, which has been proposed to be, if you will, the gold standard of measuring environmental enteric dysfunction. And that was reviewed nicely by Dr. Lauer in a previous seminar. But as you can see, stool markers for inflammation, which are the top three measures, myeloperoxidase, alpha-1 antitrypsin, and neopterin, correlated only fairly and not so significantly with the gold standard of lactulose mannitol ratio. And if you can just scan the Pearson correlation coefficients, you can see that the highest one is probably 0.39, a correlation between total immune globulin M and total IgA. Obviously, this is a non-specific relationship with markers of inflammation. So this is the challenge in our field, if you will, is that there is not the best correlation between different biomarkers of EED. This lack of specificity, I think, has hindered our ability to pinpoint which children, which mothers, which adults are suffering from this condition. Next slide. Well, our group has focused on another marker of environmental enteric dysfunction that is based on the cartoon shown here. On the left, you can see an example of a gram-negative bacteria. And if you think back to your high school biology class, you remember that bacteria are motile, and they're motile because they have a flagella at the end, as seen in the far left of this cartoon. And these allow the, for instance, E. coli to swim through water and other fluids. In addition, there is a bilipid cell membrane, and an important component of this is shown as lipopolysaccharide, a complex component of both fat and a carbohydrate. And this lipopolysaccharide, another name for it is endotoxin, and the classic experiments from the 1950s and 60s confirm that when a gram-negative bacteria such as E. coli is broken apart or is attacked by the body's immune system, release of that endotoxin, as opposed to exotoxins, but release of that endotoxin leads to a very pro-inflammatory state with release of cytokines and, in extreme cases, septic shock. So we took advantage of the fact that antibodies to flagellin and lipopolysaccharide can be measured in the bloodstream, and preliminary data suggested that people with increased permeability, less well-structured and functional gastrointestinal tracts had higher concentrations of these antibodies in their bloodstream. Next slide. The first paper that we published using serum antiflagellin and anti-LPS antibodies was performed in the setting of a randomized trial we had recently concluded in peri-urban Tanzania. Next slide. And on this table, we pointed out that regardless of which antibody we were measuring, be it antiflagellin IgA or anti-LPS IgA or IgG of these two components, next slide, there was a substantial doubling of the risk of subsequent underweight. So these antibodies were measured close to birth at 6 weeks of age, and the children were followed until 18 months of age. And what you can see in the red box is that compared to people in the lowest quartile of any of these EED antibodies, children in the highest quartile, the fourth quartile, if you will, of this exposure, had approximately doubling of the risk of developing low weight for age z-scores over the course of follow-up. So this was quite intriguing and led us to consider whether evaluating this inflammatory response that's specific perhaps for the gastrointestinal tract might be related to other health outcomes. I will say that reviewers of this paper also pointed out that because these antibodies might be related to more systemic inflammation, a topic we'll talk about later today, the question is, were these specific for this effect, or was this really reflective of a generalized inflammatory response? Next slide. So we basically did another experiment, and this is one that Jackie Lauer led. This was a birth cohort, which we performed with colleagues in Uganda and with support from the Nutrition Innovation Lab. So this birth cohort was performed, and at 6 months of age, a cross-sectional analysis of multiple markers of environmental enteric dysfunction were collected, including hemoglobin and measures of systemic inflammatory responses, including AGP and CRP measures of that. At 6 months of age, I'll point out that this rural cohort had a 35% stunting prevalence and a 53% anemia prevalence. And controlling for these measures of systemic inflammation, markers of EED were still quite highly correlated with both length for age z-score as well as hemoglobin. And the hemoglobin correlation shown in the figure on the right shows a weak but statistically significant relationship such that with higher concentrations of lipopolysaccharide antibodies, you can see lower concentrations of infant hemoglobin at 6 months of age. Next slide. Dr. Lauer also performed another study, this time in Mukono district in the central region of Uganda, which is about 20 kilometers east of Kampala. among 258 women who were pregnant and attending clinic for the first antenatal clinic, Dr. Lauer and colleagues performed multiple measures of environmental enteric dysfunction, including the measure that I noted previously as the gold standard, the lactulose-mannitol ratio. Interestingly, the lactulose-mannitol ratio showed no relationship between important birth outcomes in these pregnant women, including gestational age duration or length for age Z-score at birth. However, and as shown by the graph, the scatter graph again on the right of this figure, you can see that with higher concentrations of lipopolysaccharide antibodies, there was a sustained and weak but significant correlation between shorter gestational age. This was the first paper that we were aware of that linked measures of environmental enteric dysfunction with important measures such as gestational age. And in the next slide, you can see, next slide, again, in the red box, you can see significant correlations between gestational age, length of the infant at birth, and length for age Z-score at the birth with a variety of different markers of environmental enteric dysfunction. Next slide. So in conclusion, a lot has been learned about environmental enteric dysfunction in low and middle income countries, but unfortunately key challenges persist. Specifically, we don't quite have a agreed upon case definition or specific diagnostic criteria. It's been proposed that because there are multiple domains of gastrointestinal function, and including decreased absorption and increased permeability, that a mix of biomarkers might be a promising path, and we're moving forward on that with different studies we are setting up. Next slide. The idea that important wash interventions and EED are related seems obvious to some, but indeed a number of wash intervention studies have been performed without clear improvements in markers of EED or childhood growth. Next slide. But I think it's clear to say that there is a relationship between EED and linear growth in infants and young children. And we're interested in partnering with you and other colleagues to find out how EED might affect pregnant women, how it might affect birth outcomes, micronutrient deficiencies, and other forms of undernutrition. So we can help, next slide, children like this grow up to be as healthy as possible. Thank you very much.
Thank you, Professor Degan. I think the key message here, it is what we need to look at is not only the safety of the food, but also the cleanness of the environment and the safety of the water. So it is looking at the micro and the microenvironment surrounding the children and within the children and their families and household. That is very impactful message that we need to work at a comprehensive packages of intervention in order to achieve better nutritional outcomes. Thank you so much. Now our next speaker, Dr. Ghosh, will speak about mycotoxin, microbiome, and environmental dysfunction. Again, Dr. Ghosh, over to you.
Thank you, Dr. Kaplan, and it's my honor to be speaking with all of you today and following Chris Duggan's fantastic presentation. So I'd like to go to the next slide, please. So just as a reminder, I mean, this is a conceptual framework around the intergenerational elements of stunting, where you have this particular framework was developed by Andrew Prendergast and Jean Humphreys. And I think all of you are very familiar with this, where you have the effects or risks that are encountered by women in preconception and conception move over into outcomes in early life, but also in later life, and that this is a cyclical process. So within the Nutrition Innovation Lab, we've tried to focus in, and I think Chris has highlighted some of the studies that have been conducted by our graduate students and postdocs, such as Jackie Lauer. What we've looked at is that green, the area within the green markers, and I'll ask Grace to please click on the next slide. Thank you. And we've looked at risk factors in pregnancy, including aflatoxins and other mycotoxins. We've also looked at EED and inflammation, some of which Chris has already presented. Next, please. And their correlations and their relationships with birth outcomes. And I think we're seeing a picture emerge, but as Chris has already outlined, that this is a pretty complex picture that emerges with respect to these different risk factors and the birth outcomes. Next, please. We've also looked at early life and up to 5 years of age at risk factors such as aflatoxins and other mycotoxins, EED inflammation, as well as assessing the microbiome in children who are moderately malnourished, and assess these risk factors relative to outcomes, including anthropometry, as well as recovery from malnutrition, particularly moderate acute malnutrition. Next slide, please. In a sense, and as this has already been pointed out by Dr. Kaplan, we've been able to conduct studies across different countries in Sub-Saharan Africa and South Asia and Southeast Asia as well. And some of them have been cross-sectional studies, like the study that was conducted in Timor Leste in Mozambique. The Uganda studies that Chris has already outlined gave us an opportunity to collect longitudinal data, not just only on diets and anthropometry, but also on specific EED markers, as well as aflatoxins. We've also been able to collaborate with colleagues at Cornell on one of their longitudinal studies called the PRENAP study, which allowed us to assess aflatoxins relative to presence and absence of HIV infection in pregnant women and the effects of those in early infancy. And in Nepal and Sierra Leone, we've been able to conduct studies that have allowed us to not just assess aflatoxins, but also mycotoxins and the microbiome relative to risk of EED. So for the purpose of this presentation today, next slide, please. I'm going to try to ascertain, based on what we have as data, some newly emerging analysis, as well as some analysis that has been presented by one of our other graduate students, Akriti Singh, for her work in Sierra Leone. I'm going to try to bring the elements of mycotoxins, EED, and microbiome. And as indicated by Ahmed, I think that we want to sort of put forward what are the implications with respect to future research that is relevant for policy and programming. So for the purpose of this next series of slides, I'm going to focus on the AFLA cohort study in Nepal and the 4-food study in Sierra Leone. The AFLA cohort study was supported by USAID Nepal, as well as USAID Bureau for Resilience and Food Security, and focused on assessing aflatoxin exposure in early pregnancy through early infancy up to 2 years of age. We had the opportunity in the study when the infants were around 18 months of age to add in assessments of other mycotoxins, as well as an assessment of the LM ratio, which, as Chris has already pointed out, is used as a sort of gold standard for measurement of EED. Similarly, the 4-food study, the Nutrition Innovation Lab was able to work with Akriti Singh, who implemented a component of the 4-food study for her dissertation. And this 4-food study is a study supported by USAID Food Aid Quality Review. So it's a separate outside the Nutrition Innovation Lab, but we were able to collaborate with FAQR and implement a small component of the 4-food study around EED and the microbiome. Next slide, please. So before I go into some of the results that I want to present, I think this is some of the questions that have come up from the registrants, as well as come up in some of the chat in the chat box right now, like what is the possible mechanism by which mycotoxins could affect or lead to impaired growth. So this particular framework has been developed by Laura Smith and colleagues at Hopkins and was published in 2012. And essentially what you have in the red boxes are 3 mycotoxins. You have aflatoxin, you have DON or deoxynivalenol, and fumonisins. And so these are the 3 mycotoxins that are implicated in impaired growth. And what Laura lays out is a very nice framework of how based on literature, existing literature, what could be the proposed way in which these mycotoxins could affect growth. And essentially what you see in the yellow boxes are, what you see in the yellow box is the process of environmental enteropathy or environmental enteric dysfunction. So in a sense, what the authors are saying is that it's very possible that these mycotoxins affect growth and development in infants and young children through the process of affecting gut health and gut integrity, which is sort of essentially defined as the process of EED, if you will. Next slide, please. In terms of sort of expanding that further, this was developed by Akriti Singh for her dissertation, and sort of she expanded that sort of diagram as well as adapted from another diagram developed by the SHINE trial and colleagues at Johns Hopkins, in which sort of in addition to aflatoxins and other mycotoxins, you could be exposed to poor wash and environmental contaminants, and that could lead to changes in the microbiota profile, which then leads into changes in the small intestine that are similar to what has been described with EED. And that in turn translates into poor absorption of nutrients, systemic inflammation, which leads to increased nutrient needs and ultimately impaired growth. Of course, these are conceptual pathways and frameworks, but these are good guides for us to assess and understand using either existing data or conducting studies to ascertain if any of these, which part of these pathways we can follow. As Chris has already mentioned, I want to sort of highlight the fact that EED is a process, and each of these red boxes you can see could have different markers that can be used to assess each of these boxes. And this was something that Akriti highlighted in her webinar that was conducted around the same time as Jackie's, and I welcome you all to go and look on the website for those webinar recordings. That essentially what you're seeing is the lack of correlations that might be observed with the EED markers could be a function of the fact that we are looking at a process and a very complex process. And not all markers are measuring the same red boxes, if you will. Next slide, please. So let me move on to some of the sort of key findings. I have presented some of the descriptive statistics around mycotoxin exposure in young Nepali children, 18 to 22 months. This is data from our AFLA cohort study, but today I'm able to take it further into multivariate analyses. But just to sort of remind for those of you who were on the previous webinar and for those of you who are new, we assessed mycotoxins, which include aflatoxin, ochratoxin A, fumonisin, B1, and deoxynivalenol. And these are all different mycotoxins that are that essentially can exist in the food system in different foods. Aflatoxins and fumonisins and DAWN usually come in on maize, whether food or feed. Ochratoxins are present across all cereals and cereal grains. So they're pretty prevalent, if you will, across the food system. And each of these mycotoxins have different effects, but most of them have been implicated in retarded growth or impaired growth. At least 3 of them are known carcinogen and 1 is a suspected carcinogen. So that's also something very important to highlight here. Now in this slide, next please, what I want to highlight is the fact that we measured in serum, we measured aflatoxin and ochratoxin in the serum of these Nepali infants and fumonisins and DAWN in the urine. And we found very high rates of detection. So what you see in the parentheses that has been highlighted is the detection rate. That is there is detectable level of this mycotoxin present in this sample. And whereas DAWN and aflatoxins were about 85 to 87%, all the samples that were assessed for ochratoxin and fumonisin had detectable levels of these mycotoxins. So it's pretty prevalent, but if you look at the right side of the table, you can see that there is a very wide range of minimum and maximum values with respect to the different mycotoxins. Next slide, please. The next thing that we looked at, we were also able to collect at the same time point, which is when the infants were 18 to 22 months of age, we were able to conduct an LM test, which is a lactulose mannitol test. And here we present the correlations between the EED markers that were developed or computed using the LM ratio. With this includes the LM ratio. It includes the percentage of lactulose excreted. It is percentage of mannitol excreted, and the lactulose mannitol excretion ratio. And what we found was 3 of the mycotoxins, aflatoxin, ochratoxin A, and DAWN, had significant, very small correlations, but significant correlations. This is a Pearson correlation coefficient. Next slide, please. Using that, given that there were these relationships, we wanted to assess the individual mycotoxins relative to the LM ratio and anthropometry. So what I'm presenting here are 4 different models in which we looked at each mycotoxin relative to the LM ratio, with the dependent variable being either stunting, so that's the first column, underweight, which is the second column, and low head circumference for age Z-score, which is the third column. And what I'm presenting is all the odds ratios that were computed using logistic regression models. Now, these models were adjusted for prior anthropometric status, either length or weight at birth or head circumference at 3 months of age. We also adjusted them for diet diversity at that time, as well as the mother's education. So what you see here is that it was only the aflatoxin model where we found a significant odds ratio with children who had higher levels of aflatoxin were more likely to be stunted or underweight. So that was the only model where we found a significant association between a mycotoxin, in this case aflatoxin B1, and the specific anthropometric outcomes. None of the models was LM ratio significant. Next slide, please. We then also looked at concurrent exposure. So we ran 1 single model per anthropometric outcome. And what you're seeing here is stunting, underweight, and low head circumference. It's a single model where we inputted all mycotoxins along with the LM ratio. And what you can see here is for both stunting and underweight, none of the mycotoxins nor was LM ratio significantly associated with the risk of being stunted or underweight, but except for aflatoxin B1. Next slide, please. And so the question is, is it possible that, based on that conceptual framework, the reason why we are not seeing these relationships is because these mycotoxins are mediating through the EED pathways. Now, while unfortunately we don't have other markers of EED, we did have, and we had only the LM ratio, what we thought was we could look at individual mycotoxins and the LM ratio as the dependent. So we ran 4 different models, and these are linear regression models, and you can see that what we find is ochratoxin A and deoxynivalenol or DAWN were the 2 mycotoxins that were positively associated with LM ratio, indicating that the higher the level of the mycotoxin, the higher the LM ratio, which indicates a higher sort of permeability, intestinal permeability, and higher impaired absorption, i.e. in higher risk of EED, if you will. So what that's telling us is that at least in this sample, we're seeing 2 of the mycotoxins being significantly correlated with LM ratio, and it's very possible that they're mediating, that their relationship to growth is through the EED process. It's possible, right? Next slide, please. So the next question really is, what do we know about EED vis-a-vis the microbiome? And this is, I'm just giving a snapshot of some of the findings that were presented by Akriti Singh, in which she looked at EED microbiome and moderate acute malnutrition in Sierra Leone. It would have been interesting if we had mycotoxin data for this sample as well, but we don't. So I'm just presenting this to sort of get us thinking about the microbiome EED, and then where does that take us with respect to mycotoxins? So what did Akriti find? So Akriti assessed EED using many different markers. She used LM ratio, as well as she used fecal mRNA markers, and she used 3 different fecal protein markers. And what she found, depending upon what marker she used, she found that high inflammation and permeability were very negatively associated with length for age Z-score and weight for length Z-score in children with MAM. And this was before they were exposed to the treatment foods, which was part of the 4-food study. She also found that through the period of treatment, that children who had high intestinal permeability when measured at baseline, they had lower length gain and lower weight gain. And finally, that children who had lower intestinal permeability were more likely to recover from moderate acute malnutrition. So this sort of emphasizes the issue that it's not just the treatment with foods is going to help, is that cleaning the environment is going to be critical for these kids to recover from moderate acute malnutrition. Next slide, please. Next. So the next thing that ACUTE looked at in her fecal samples was she did a sequencing to assess the microbiota and the microbiome composition of these kids with moderate acute malnutrition. And what she found was, next please, that the gut microbiota of the children with MAM were very enriched with inflammogenic taxa. So on this figure that you're seeing, where you're seeing the red bars, those are the inflammogenic bacteria, and those were present in very high levels in the kids with MAM, and that the alterations in gut microbiota were very associated with MAM, and it was also associated with inflammation as measured using different EED markers. As I said, we would have loved to have an assessment of mycotoxins relative to the gut microbiota as well as gut inflammation with the EED markers, but we don't have the data. And that's really where what I'd like to see us thinking about in the future, that do we know about how mycotoxins interact vis-a-vis the microbiome? And does a varying or variable microbiota affect the sort of, does it remove the effects of the mycotoxins? So that's really a future area of research that needs to be explored. Next slide, please. And what's very interesting is that there is a recent review on this topic, particularly on mycotoxins and the gut microbiota, because to a certain extent in animal research, it has been shown that when you have an environment that is enriched by probiotics, so you have sort of, if you will, good bacteria, it is known to biotransform mycotoxins to less toxic metabolites. And so that sort of highlights the fact that having a good microbiome might be supportive and might be protective of the effects of mycotoxins, something that needs to be examined within human studies. On the flip side, if you have changes in the microbiota composition, as ACUTE observed with respect to MAM, it is possible, as noted by this review, that there could be a consequence when there is mycotoxin exposure, where you could have changes from a good microbiota to a bad microbiota because of the antimicrobial properties of the mycotoxin itself, or the toxic effects that the mycotoxins might have on the epithelial and the immune cells of the gut. So essentially the flip side of this is that if you have a very good microbiome, you might be able to negate the effects of mycotoxins, but that if you have excessive exposure to mycotoxins, it might change your microbiota composition. So that's essentially what in a nutshell that needs to be explored and understood further within humans. And why is this important? Because we all know that we're not living in an enclosed environment where only one risk factor matters. What we are seeing is that often there are multiple risk factors that are going to be critical and that need to be addressed. And I think I wish I had used Chris's slide from the Lancet series, where you could say that some of the interventions are, even if you have those 10 at 99%, they're going to only affect those specific outcomes up to a certain degree. And so that's why it's really important for us to consider the future of mycotoxins and microbiota. Next slide, please. So I think I'm really way out of time here, but I just want to conclude that we've found some very interesting things with respect to multiple mycotoxins in Nepali infants. we're seeing some very interesting findings around EED and how it modulates recovery in MAM. And there are reviews that are indicating the need for assessing the bidirectional relationships of mycotoxins and gut microbiota. Next slide, please. So we really think that we should be looking at assessing interactions between these environmental toxins and microbes and the process of EED in the gut microbiota. And these are complex assessments, but they need to be done because it's going to help us drive future policy and programming within the context of food systems, agriculture, nutrition, and health. Thank you very much. And I'm going to pass this back on to Ahmed.
Thank you so much, Dr. Ghosh. That is a very important message that you left us with, that factors contributing to child malnutrition, and they are not one factor. There is multiple factors, and you find some, children of, especially the population that you study, they are exposed to bad environment in terms of poor hygiene, as well as unsafe food, as well, which is contaminated with multiple things, including potentially several mycotoxins, different kind of microbes. So that really bring the importance of the message that the development community must work together in a unified voice, advocating for improved food safety practices, linking food system with the health systems in order to really deliver on improving child nutrition and outcomes. Finally, we will move to Dr. Patrick Webb. Professor Webb will talk about what are the programmatic implications for all of this knowledge that we have. Oh, Professor Webb, over to you.
Thank you, Ahmed. Those were two really important presentations. And I'm going to actually quite briefly bring us more to the level of programmatic and policy thinking around the implications of these things. Multiple factors, you just used that term, Ahmed. I think we've heard so far, we've heard about mycotoxins of different kinds, we've heard about EED and E. coli, we've looked at inflammation and gut microbiome. A lot of people would love to have simple answers to deceptively simple questions. You know, how does EED affect stunting? How does inflammation impact linear growth, and what can we do about it? But what we've been hearing so far is that these kinds of processes, they're not necessarily events, they're processes within the body. They may sometimes be synergistic, they may be antagonistic, they may be additive and cumulative in their effects, or in fact, one may be more important than the other. And that's really where I'm focusing in here, is that we need to consider the multiplicity of possible threats or drivers or causes of the problems that we seek to resolve. That doesn't mean programmers have to measure them all themselves through the various highly complex techniques that were presented just now, but they do need to understand through theories of change how an intervention could potentially modify these risk factors, the term that Shivani just used, and more importantly, did they? Next slide, please. Did they actually succeed in modifying risk factors? Now, I put this not expecting anyone to be able to read it. It's simply through the Lancet series and others, assessments of the global burden of disease. And that global burden, the relative contribution of elements to the global burden of disease, changes over time. And so I think one important point we're trying to put on the table here is that these different risk factors we've been talking about, EED, aflatoxin, and so on, matter differently in different places and potentially over different times. I wanted to, this is two columns, 1990 and then 2010. So 20 years later, in terms of the red, we see protein energy malnutrition, an old term for undernutrition. In 1990, it ranked 9th in the world in terms of contribution to the global burden of disease. By 2010, it had dropped to 20th place, and by 2030, it will almost undoubtedly have dropped still further. Iron deficiency anemia, on the other hand, 14th in 1990 was still 15th in 2010. And we know that anemia remains one of the great challenges that's still there in nutrition, and we're struggling to make a lot of headway. Other things got worse over time. HIV/AIDS was 33rd in 1990, climbed significantly to be 5th in 2010, may well have dropped a little in the meantime, relative to others like diabetes, 21st in 1990, 14th in 2010, and almost certainly climbing higher in the meantime. So the point here is that the relative risk of different contributions to health outcomes and nutrition outcomes, they're not static. And so in trying to understand the contribution of, say, EED to child growth requires us to understand where that sits in the context of other risk factors in any particular environment. Next slide, please. For example, well, just keep going. I forgot those. For example, so in our study in Nepal, we looked at a variety of risk factors that related to, for example, low birth weight, one of the key birth outcomes of interest, because we know that low birth weight, and in particular others, like being born small for gestational age, those are birth outcomes that are highly correlated with later stunting among children. Right? So being born with suboptimal outcomes at birth is itself setting in motion stunting, setting in motion the process of suboptimal growth. So, but in this particular sample in Nepal, risk factors for low birth weight, in a setting where, although there was widespread exposure to aflatoxin in this particular case, the levels of aflatoxin exposure were relatively low, but click first, next slide. What we see that while, next slide, next click, while aflatoxin in the blood of mothers was predictive, it was, you were more likely with higher levels of aflatoxin in blood, mothers were more likely to have low birth weight in their subsequent children. But low maternal stature and smoking were more significant in that environment. Now, what that means is not that aflatoxin in the blood is not important. What it means is that right now there are other factors that also contribute to low birth weight that need to be addressed. And smoking is a modifiable risk factor, is one that we can change immediately. We have to change in so many environments. Low maternal height, of course, is going to take longer, intergenerational, because we need to break the cycle. We need the next generation of girls to not be born with low birth weight and to grow more optimally to achieve their potential genetic height. Once those two problems have been resolved in this context, then the significance of aflatoxin in the blood, if we don't deal with it, will dominate, right? So you get what I'm saying. These factors, it's not that this one is less important. It means others are equally important, have to be addressed. But if we don't address the aflatoxin in the meantime, then its significance relative to others will grow over time. So we should address it immediately. Next slide, please. Now I'm going to talk just very briefly about some of the water issues as opposed to aflatoxin. Really, from the perspective of, in a sense, fixing the problem, right? So we've heard that from Chris that more E. coli is linked to EED in the child because of the leakiness, the malabsorption in the child's gut. And we've also seen that more EED is associated both with stunting in children and wasting. Wasting is not presented here, that's presented separately. But we have those links. E. coli is a problem in terms of EED. EED is a problem in that it is associated with certain nutrition outcomes. So what do we do? What's the fix? Next slide. Well, there's been a long-standing agenda of ensuring universal access to clean water. And I emphasize the word clean. It's not about universal access to water. It's universal access to clean water. And therein lies a problem, right? Because access to water has improved over time through a variety of interventions, through wells and boreholes, covered wells, uncovered wells, improved pipes, and so on. Next slide. What we see, though, sometimes in treatment, direct treatment of water sources, next slide, or improved or unimproved types of water access, you're still seeing roughly 60% of all kinds of the water access in this particular location in Southwest Uganda having contamination, empirically contamination with E. coli, right? So sometimes the not treated is worse, the unimproved is worse, but not by a whole lot. But my point is, even improved sources of water, treated sources of water, plastic jerry cans versus traditional plots, you're still seeing more than half of the samples contaminated with E. coli, right? So it's not just about, oh, we've fixed the problem by ensuring a pipe. Next slide, please. Sometimes it's the use and behavior of those things. Next slide, please. Next slide. In that location in Southwest Uganda, you see the two columns there of safe versus unsafe, and you see that of the piped, there were only 8 piped water sources in that particular community. Half of them were deemed unsafe in terms of E. coli contamination. Public taps, more of them were safe than unsafe, but actually 42% of them were contaminated with E. coli. Protected boreholes and protected wells, significantly more unsafe than safe, right? So what we're seeing here is that you cannot rest by dropping a borehole, covering a well, or ensuring access to a tap. That is not job done. That is just the first part of a job done. And the next slide showed that again, correlation with EED, the lactulose mannitol ratio, showed a significant relationship with length for age and weight for high Z scores. Next slide. Now, some of this can be related to, obviously, behavior. In the Uganda study, this slide relates to Ghana, but before I leave Uganda, we've not published yet, but we've been looking at the impact of a multi-sector program in various parts of Uganda, one of whose components was to improve sanitation and hygiene by teaching about hand washing behavior. And we've been we're deep in the analysis of this, and a lot of things improved through that program. A lot of things were very positive. But what we've seen actually is that over time, households most involved in the program actually reduced the frequency of hand washing. Reduced the frequency with strong SBC messaging on quality, on how to wash your hands. And one possible solution to that conundrum may be that the households heard the message about how to hand wash better, the whole 2-minute thing with soap or ash and so on. And as a result of that, wash their hands less frequently. That's possible. We don't know. We can't explain that. But these are some of the challenges that have to be faced. This is a map and a diagram relating to that map of a small township in Ghana. The orange pipes are stand pipes. They're public pipes that theoretically are delivering clean water. But there's low consumption of water at those stand pipes. The green ones further away from the river, also notionally delivering clean water, have a high consumption overall, on average, much higher. And one can, the orange line on the graphic to the left, these are the consumption levels for the 3 pipes on the right. And what was documented through this particular study is that low consumption was related to proximity to the river. Why pay at all for water if you have to pay? when it comes free from a river, right? So you have low consumption here. These communities around these, much further away from the river, made much more sense to use the sandpipe. But click next slide. What you see is that consumption level from those pipes drops during the periods of the rainy season, a bimodal rainy season. So the more rain there is, the much less consumption from the pipes there is. And again, one may say, oh, well, that's logical. They may just catch rainfall. In fact, during the rainy season, what they're doing is collecting rainfall from surface water, seasonal streams that develop during the rainy season and taking water from there. And when asked, well, why are you doing that? Normally you use the pipes and you know that the quality of water from those pipes is much higher. And the answer was, actually, we prefer the taste. We prefer the taste of the groundwater. Actually, many of them preferred the taste of the river water than the water that was coming from the pipes. Right? So the moral of the story here is that simply making clean piped water available doesn't mean that people will use it or use it adequately on a systematic basis. Next slide, please. So the relative risk of these different threats, these risk factors, is going to change on context. So really one of the messages for programming is it's not black and white. You've got to know the context. Which foods in your context are known to be high carriers of various mycotoxins? What do you know about the water quality, even of the supposedly clean water sources in your context? What are the disease parameters on a seasonal basis? Yeah, we all know this, but it needs to be reiterated, because what we're understanding through this kind of research that is pushing the boundaries of understanding is that variety of stressors, inflammation, mycotoxins, E. coli, and diseases like HIV, which themselves stimulate inflammatory processes, they are clearly interacting or not in important, but not always predictable ways. And so we need to understand more about how those interactions take place, how humans react to the stressors in their environment, and what, therefore, we as development agents of change can do to either modify those behaviors in positive ways or change the cleanliness of the setting. Access to universal clean water matters as much as access to clean food environments and clean health environments. Programs themselves shouldn't be seeking to measure everything. We're not asking people to measure blood serum, aflatoxin levels, or even lactulose mannitol tests. But theories of change do need to be much more rigorous and transparent in their assumptions. I've looked at a lot of theories of change, programmatic design over many, many years. And I don't see this happening. I see far too many assumptions and a fairly hand-waving approach to the links between boxes. And what we need to understand here is the dynamics and the potential net interactions among these various stressors. Of course, knowing more matters, field-friendly metrics. There are better, quick ways of assessing proxies for E. coli or cleanliness of the water using watercolored bags. There are proxies for assessing food, mycotoxins in food as opposed to in serum, but we still need to go a long way more in that direction. Field-friendly techniques are going to help us all a great deal. But last message, these stressors matter hugely physiologically among nutritionally vulnerable populations. They're not linear relations between mycotoxin and stunting, but the processes that contribute to suboptimal growth of children, adolescent girls, and then pregnancy outcomes are all somehow related to these stressors. And we do need to know how they interact and what we can do to change that situation. I'll end on the terrible note that I just heard in the news that the UK, my government, has just announced it's going to cut funding for water and sanitation interventions in developing countries by 80%. in the coming few years, which I think is shameful and based on what we've just been seeing, extremely short-sighted. So I hope we can change that around in the near future. And thank you. Back to you, Ahmed.
Thank you, Dr. Webb, and appreciate the programmatic messaging that you left us with and the complexity of the issue. But I really appreciate the one message that you mentioned, the importance of transparency of the theory of change and to have it built on testable assumptions, not on assumptions that could not be validated or tested. This is very critical to establish and build programs, especially if you want to go back and test some of these assumptions in a researchable methodological way. We have received, as you can see, a lot of questions through the live Q&A, but also we have a good list of questions that have been submitted during the registration period. And about 22 questions have been answered already by the speakers. Thank you so much for that. I will ask some of the questions that from the live question, as well as some questions from this question that have been submitted. Let's start with one of the easy questions. In terms of the aflatoxin, and this might be for you, Shibani, how long does aflatoxin remain in the gut and the serum of children under 2 years? And does it last long enough to influence the composition of their gut microbiota?
Yeah, that's a very good question. I can say that the serum aflatoxin, which is what we use as the measure or the marker in our studies, has a half-life of 90 days. If you measure aflatoxin, a metabolite of aflatoxin AFM1 in the urine, that's usually a much faster turnaround. It's 24 hours. So it really depends on the marker you're using with respect to exposure, whether it's a serum marker or a urine marker. In terms of the absorption of aflatoxin, and is it sufficiently long enough in one instance in the gut, I don't know the answer, but I do know that what we find in the populations we are working in is there is consistent chronic exposure to aflatoxin. So even if it has a very fast transition in terms of absorption through the gut, what I can tell you is that they are being exposed to aflatoxins on a daily basis. And that there is, there might be others on this webinar who might be able to answer specific question on gut, absorption of toxins like aflatoxin in the gut.
Thank you, Dr. Ghosh. And a question for Christopher related to the environmental enteropathy. How fast the condition will be reversed if you improve the environment, food and water, etc. And is there a long-lasting damage?
It's a great question, and the short answer is we don't know. The early studies in this field suggested that when adults left an environment that was correlated with high enteropathogen exposure, that their absorption of nutrients improved, so leading people to think that it was a temporary issue. But of course, most people living in low and middle income countries can't just exit their environment. That's where they live. And so chronic exposure likely is correlated with chronic effects. Whether short-term exposure leads to chronic effects is hard to know, but it's an area of great interest, because it seems early studies that, and some of which we've done, have suggested that, for instance, there depending on what marker of EED is used, there are, for instance, neurodevelopmental effects of early infancy EED.
Thank you. A question submitted during the registration period, and I'm going to ask it to the panel. And this is related to climate change impact on. And so the question submitted from Principal and Food Security Officer at the Ministry of Animal and Agriculture, Industry, Fisheries in Uganda. The question: how can government mitigate the effect of aflatoxins on children growth under condition of climate change? But I want also to expand this to look at other factors that could lead to negative impact on child growth and environmental condition, environmental enteropathy, to look at how you anticipate also climate change could impact the spread of microbial contamination or contaminant that could lead to a condition like an EED and others. Over to you. Who wants to start it? Okay, come in.
Well, okay, I'll jump in, I guess. Very forward-looking and good question, obviously. There's very likely to be expansion of mycotoxin contamination threats into new geographies with climate change, and maybe intensification of those threats in areas where they already are, simply because of the temperature, humidity, rainfall effects. And therefore, attempts have to be made to prevent the problem from growing, right? And that means improved seeds, improved management on farm, but especially improved storage and improved processing, and therefore knowledge and awareness, not just of the smallholder farmer, but of the marketplace. And that means governments have to play their role, not assuming this is just something that farmers have to deal with. Regulations, testing, of food testing and safety standards, all are going to matter a great deal in future. So you combine that with the potential lowering of water tables and water stresses, which are already manifest in many low-income countries. That combination of water stress and food stress is pretty scary when you look down the future. So we better start acting on it right now to prevent the worst. Over.
Thank you, Dr. Webb. Any other of the speaker would like to add to it, or should we move to the next question? I have an interesting, several other interesting questions. One of them I really want to pick on it came in terms of looking at thinking about environmental enteropathy and impact on gut microbiome, as well as potentially aflatoxin impact on gut microbiome. What are your thoughts about adding pre and probiotic to diets along the other wash interventions, as well as the other nutrition packages, to support the health, the assembly of a healthy microbiota in the gut? Any thought on that? Christopher, you want to take a stab with?
Oh, sure. I don't mind saying that I'm very much a probiotic skeptic. There have been many, many different trials evaluating probiotics, and I always think back to the composition of human milk, which is very rich in prebiotics, right? The oligosaccharides in human milk are in part responsible for the development of the intestinal flora, a healthy, diverse intestinal flora in the colon of breastfed infants. So I think the most important role of prebiotics are the naturally occurring ones that are included in human milk. It does beg the question, of course, of this really cutting edge idea that Jeff Gordon and Tamid Ahmed and others have proposed, which is, are there certain patterns of diet that can be used for the treatment of malnutrition to improve weight gain and engender a better, if you will, microbiome pattern? And I think I, in the chat, had sent some references for some recent New England Journal papers on this very topic. So it's a very exciting area, but I think still we have a lot more to learn.
Thank you, Chris. Another question to the speaker from where the audience, or Sarah McClung, was interested to hear more about the policy implication based on what evidence we currently have. More specifically, any thought on conceptualizing the basic or the most basic packages of intervention that consider potential mycotoxin in nutrition linkages, what that could look like? And I think that question to you, Patrick.
Well, but also Shabani. I think it's so easy to say, well, of course we have to have multi-sector integrated programming that ensures that the water is clean and keeps children or infants away from livestock and chickens in particular, and ensures access to clean food, and appropriate hand washing and sanitation environment, right? I mean, none of these are actually quite that new, but the evidence, scientific evidence on just how important these things are and how much they constrain investments from having the impacts you'd like to see, suggests that we really need to do a lot better, and test and measure what we're doing to ensure that it is really actually achieving the outcomes that we assume it's achieving. So, I'm not suggesting much more testing by programmatic or policymakers of the drivers necessarily of these challenging interactions, but we all call for evidence-based programming, right? Evidence-based policies. But we absolutely need evidence at the other end that those policies and programs based on evidence are doing what we expected them to do, and if not, why not? And how to make changes accordingly, right? So cost effectiveness, benefit cost analysis, and testing of hypotheses and assumptions in programming, I think are all really quite important and need to play a bigger role in programming M&E systems and the investments that donors make. Over.
Ahmed?
Go ahead, Shibani.
So I think I have actually from the previous question as well as this one, I wanted to just make two comments. One is that I think I would say that we're at that point, we're not at the point where you can say that this is the intervention that was going to support a healthy biome and therefore we're going to remove EED and that's going to translate into better growth. I think there's a lot more in the space that needs to get done, whether it's on the metrics within the context of EED, whether it's in the metrics of what is a healthy microbiome, and can we even sort of sequence the biome itself. So there's a lot on the upstream end of research around this topic that still needs to get done. So there's no sort of, yes, give a probiotic or give a prebiotic. The other point that I wanted to make was that in terms of package of interventions, actually what we do know is that there are keeping a clean food system, having a clean environment, the good diet. So there's the basic tenets of these interventions do exist. But to Patrick's point, I think we don't do enough in understanding whether those policies that are targeting these specific tenets are actually translated into action and if that has an effect. And I think so there are two different ends to where I'm looking at this. One is what still needs to be done on the upstream end and research to sort of push new innovative interventions. And two is what needs to be still done on the existing package of interventions and approaches. So that really, so for me, there's a duality to this. There's no one single way of going with these two questions.
Thank you, Shibani. And another question related to this from Dr. Debendrat here from Nepal, USAID Nepal. He's asking about the linkages and implication you would see between food system, health system, and nutrition policies, and overall programming based on the neurogenerated. And I think this is a question I want to relate on the whole panel because each one of you bring different perspective from health and nutrition system as well as food system point of view. Anyone would like to start?
Patrick?
No, not me. I actually didn't catch the question.
Sorry, the question is implications of the data that we have or the evidence that we have so far.
Yeah. And
on the linkages or potential linkages between on food systems, health system, and nutrition policies.
Well, I mean, I don't have any great answers except to point out that obviously it's to focus too much, for instance, on the Ministry of Health in low and middle income countries and not thinking about other ministers, including agriculture ministers, environmental health ministers, and others whose policies have a direct effect on the problem is not the way to go. And I think what you've heard, not just from the panelists, but from the questions in the audience, really this idea has to be a holistic approach. To go back to my first slides, if we make all of the interventions that we know are effective, we're only going to reduce childhood stunting by 36% or so. So indeed, a trans-governmental approach to these problems is really important.
I think, mycotoxins are let's see it as an opportunity for encouraging that kind of dialogue, right? As we've seen in several countries, aflatoxin threats have been known and tackled by ministries of agriculture primarily for decades, for very, very long periods, because they are known to affect livestock productivity and survival and so on. And there are very strict limits, regulations through codex on the movement of contaminated foods across borders, right? So it affects trade, right? So the ministers of agriculture, ministers of trade have long understood the threats to, from an economic perspective, of aflatoxin. A lot of this new research is bringing on board the medical and public health perspectives to understand that this isn't just about dying of cancer through eating contaminated foods. There's something here that affects basically the physiology and birth outcomes and children's growth and development, right? So what this does is serve as a reminder of the interconnectedness of development activities, or how they should be. And there are countries like Nepal, like other parts of the world, where they're making efforts to better link, interdigitate actions across sectors, across ministries, in ways that can do better than they've done in the past. So I think these kinds of findings matter, but you have to get these findings not just to scientific journals, you have to get it into the hands of the policymakers and to the donors who can make a difference.
Thank you, Patrick. I think we are almost at the time, and I just want to say before we hand it back to you, Kate, to closing out, this whole presentation and looking at the complexity of the factors that contribute to the nutritional outcomes that has several linkages to food safety, the quality of the diet as a whole, the environment surrounding, the water linkages. It really bring it back to the importance of how much we really want to, we have to think on a systemic level, rather than an individual household or personal level or single intervention. As Patrick, Christopher Shibani mentioned, we need to look at it as a holistic approach from all angles and what could be the most effective packages. Personal, maybe not, also not looking at one size fits all, because one size fits all does not really work everywhere. And that's why we have to be thinking more broadly. And again, I want to emphasize one of the points, the importance of tested or testable, potentially to be validated assumption to be built that should be used. And finally, I want to say, this gives me the point to say, unsafe food is not only undermine the children's well-being, as we have seen, it also threatens their lives. We can't afford not to invest and to look at food safety from all aspects, what's microbial or mycotoxin or others. Unsafe food impede our ability to achieve nutrition and other development targets. The diarrheal disease are #1 cause of children under 5 death. And up to 70% of diarrheal diseases are caused by unsafe water and food. So we can't just do nothing. And Christopher put it nicely, if we implement 90% of the nutrition-specific intervention or proven intervention, we're achieving 35 efficacy. With that, I thank all the audience. Thank you so much for being with us for about an hour and a half, and I will hand it back to you, Katie.
All right, well, thank you so much. And thanks to the presenters for the wonderful presentations and to all the panelists, or sorry, the participants for a great discussion, and hope you have a wonderful rest of your day.
Thank you all. Thank you, Ahmed.
Thank you, Ahmed. Thank you, everybody.
Thank you.